With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, let us see the question. The given figure shows three velocity substrate concentration curves of an enzyme reaction. What do the curve X, Y, and Z depicts respectively? So option A is X is normal enzyme reaction, Y is competitive inhibition, and Z is non-competitive inhibition. Option B is X is enzyme with allosteric modulator added, Y is normal enzyme, and Z is competitive inhibition. And C is X is enzyme with an allosteric stimulator, Y is competitive inhibition added, and Z is normal enzyme reaction. And option D is X is normal enzyme reaction, Y is non-competitive inhibitor, and Z is allosteric inhibition added. So this is a curve which is given. This is a graph which is given. On x-axis we can see substrate concentration. On y-axis we can see initial velocity, and there are three curves X, Y, and Z. We need to find which curve represents which reaction respectively. So the rate of the reaction is directly proportional to the substrate concentration. Rate of reaction is directly proportional to substrate concentration. Substrate concentration, and enzymes are the biocatalysts which increases the rate of reaction. So enzymes are the biocatalysts. These biocatalysts increases the rate of reaction. So let us learn in brief about this enzymatic reactions. So initially, when there is a presence of an enzyme, on x-axis we can see substrate concentration, and on y-axis we can see velocity of the reaction. So in the presence of an enzyme, since enzymes are biocatalysts, they increase the rate of the reaction. So however, when the substrate concentration is at a higher concentration, when substrate concentration is high, the enzyme will be saturated and then the reaction will be stabilized. So initially, the reaction in the presence of enzymes, Vmax is attained at a lower substrate concentration, and then the reaction is stabilized because enzymes get saturated at higher substrate concentration. So in the presence of an enzyme, the normal Vmax is attained at a lower substrate concentration. So this is Vmax. So this reaction is in the presence of enzyme. So at the lower substrate concentration, Vmax is attained and then it is stabilized. And the next condition is this condition 1, in the presence of an enzyme. That is a normal enzymatic reaction. And the next condition is in the presence of a competitive inhibitor. The competitive inhibitor, in the presence of a competitive inhibitor, there is competition for the enzyme between the inhibitor and the substrate. So the inhibitor decreases availability of enzyme to the substrate. So there is competition between the substrate and the inhibitor. So reduces the availability of enzyme, reduces the availability of enzyme. So in such conditions, this is substrate on x-axis and y-axis velocity, and this is a Vmax. So in such conditions, Vmax is attained at a higher substrate concentration. Vmax is attained at a higher substrate concentration and then it is stabilized because there is competition for enzyme between the substrate and the inhibitor. So Vmax is attained at a relatively higher substrate concentration. So this is the substrate concentration at which Vmax is attained. So in the presence of competitive inhibitor, Vmax is attained at a higher substrate concentration. And the next condition is presence of a non-competitive inhibitor. Third condition A is presence of a non-competitive inhibitor. In non-competitive inhibitor, there is no competition between the inhibitor and the substrate, but it affects the inhibitor will reduces the activity of enzymes. Inhibitor reduces the activity of the enzyme. So therefore, Vmax is not attained in such condition. Vmax, and this is substrate on x-axis and y-axis Vmax. So in such conditions, Vmax is not attained in the presence of non-competitive inhibitor. So these are the different conditions in an enzymatic reaction. So let us see the statements which are given. So X, where Vmax is attained at a substrate concentration relatively lower than Y. So X is a normal enzymatic reaction. X represents normal enzyme reaction. So normal enzymatic reactions, and Z and in Y, as we can see, at a higher substrate concentration Vmax is attained. So this Y represents in competitive inhibitor. Competitive inhibition in the Z graph. Y graph, and Z graph represents a non-competitive inhibitor because Vmax is not attained. Non-competitive inhibitor. So the answer for the question is option A. That is, X is a normal enzyme reaction, Y is a competitive inhibitor, and Z is a non-competitive inhibitor. Hope it's clear. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.